Okay, good morning, afternoon, and evening to everyone. I'm Cosmo Camiso, your host for today. Together with our moderator, Dr. Swasti Harikaran, we'd like to thank you for joining us for our latest installment of our Focus on Seminar series at Center, where we pair a rising star with a opinion leader. Today's topic is pancreatic cancer, but you will see that the work that these two amazing scientists do in their labs reaches far beyond a specific tumor type.
Right, thank you. I hope everybody can hear me, and I want to thank everybody, the organizers, for the opportunity to present. Let me see if I can get my laser pointer thing. Well, so these are my disclosures. So as I'm sure everyone here knows, MYC is a transcription factor. Here's the pointer thing, let's see. Pen. Oh, that's not what I want to do, sorry about that. That's not working either. Okay, hopefully you can see my arrow. So MYC is a transcription factor. It binds E boxes in the genome, and when it does, it recruits chromatin remodeling factors that result in the phosphorylation of post-RNA polymerase, leading to its elongation. Recent insight into MYC has revealed that it's really a master transcriptional regulator, controlling pretty much all genes in the genome, and it can bind to any open, actively transcribed genes MYC is found to be bound to. And at high levels in cancer, it can also invade into more closed chromatin to induce the opening and new transcription. And through these global activities in the genome, MYC controls virtually all of the hallmarks of cancer. And this includes cell intrinsic aspects of tumor cells, such as cell growth and division, immortalization, reduction in differentiation status. But it also controls cell extrinsic factors, like in the tumor microenvironment. Tumor cells with high MYC will have, the microenvironment will have increased angiogenesis and immune suppression. So recently, Dr. Evans, Gerard Evans, published a very nice paper that we were invited to write a commentary on that really shows the way in which MYC can control all of the hallmarks of pancreatic cancer. So in this model, KRAS is mutant and on, but it really only drives ADM and a little bit of penin, as other people have shown. But then when you switch on MYC using a tamoxifen-inducible MYC-ER fusion protein, the tumors rapidly progress to PDAC. And along with that, you get a large expansion of the tumor microenvironment into a desmoplastic, immune-suppressed, highly aggressive microenvironment with this PDAC. And then because he has this dox or tamoxifen-inducible MYC, he can stop giving the mice doxycycline or tamoxifen, and the MYC turns off. And then what's amazing is the PDAC disappears, there's death, and you're left with only some pannins and ADM. And at the same time, the desmoplastic tumor microenvironment also goes away. So this really speaks to the role, both cell intrinsically and cell extrinsically, of MYC in driving cancer. So my lab, over many decades, have been studying post-translational regulation of MYC. And we know that newly synthesized MYC can be phosphorylated through the RAS-RAF-MEK-ERK pathway on a conserved serine 62 site. And when MYC is phosphorylated at serine 62, it's a substrate for proline isomerization at proline 63. And this accelerates the rate at which MYC binds to target genes. This phosphorylation event, however, also primes a subsequent phosphorylation event at threonine 58, which is held in check when by PI3 kinase AKT signaling that also is usually downstream of extracellular signals. And so by holding this in check, MYC is more stable. But then as signals die down, MYC, this phosphorylation recruits PIN1, the proline isomerase, yet again for a second isomerization event that then switches proline 63 back to trans. This makes MYC a substrate for the PP2A phosphatase. This phosphatase dephosphorylates the serine 62, this stabilizing activating site, resulting in phosphothreonine 58 being recognized by the SCF FBW7 E3 ubiquitin ligase and degraded. In cancer, this part of the pathway is turned down and MYC is left in a more stable, more highly phosphoserine 62 form, and it's more transcriptionally active and can drive cancer. And this is an example of a phosphospecific antibody that we have, and we had one a while back that was a polyclonal that worked beautifully, but now we finally managed to generate a monoclonal that I'm really excited about because it works on FFPE tissue from both human and mouse. And you can see here high levels of phosphoserine 62 in PDAC. So in more recent publications, we've really detailed this post-translational, dynamic post-translational control of MYC more. And I'm not going to show you those because it's been published, but I'm going to give you a schematic. And the reason I want to show you this, it's a little complex, is really to emphasize the temporal nature of post-translational modifications of MYC that really allow it for its dynamic activation and inactivation. And so what I'm showing you here is that signals from the tumor microenvironment stimulate serine 62 phosphorylation and the recruitment of the PIN1 proline isomerase. And this facilitates the rapid recruitment of MYC to DNA binding sites where it recruits the co-activators, as I said. However, subsequent phosphorylation by threonine 58, we've shown kicks MYC back off of the DNA, and this sets it up for ubiquitin-mediated degradation. But it's this bind and release that is important for the release of pause polymerase. So it's this cyclic DNA binding that occurs. And then MYC in normal cells would be degraded. And we've actually shown that there's a mixture of ubiquitin and sumo modifications on MYC that target it for degradation. But in cancer cells, there's these other factors that get upregulated, such as USP36, which is a deubiquinase to take off the ubiquitins, and SENP1, which is a desumulase, which desumulates MYC. And this leads MYC back to a ground state where it can be activated again and recycled. And that's one of the reasons why MYC is more stable in cancer and more active. So as I said, sequential phosphorylation leads to cyclic oscillatory DNA binding, and this is important for the release of pause polymerase. So more recently, we asked whether, is there not only a temporal dynamic to MYC control, but is there a spatial dynamic? And this was work we collaborated on with Yuka Westermack, where we discovered that phosphorylation of MYC at serine 62 is a form of MYC that seems to be accumulated on the periphery of the nucleus. And this was really interesting for a couple reasons. One is that a long time ago, 1991, EM studies had shown, it's really hard to see, but basically in these little nuclear pore structures, there were gold granules, which showed electron microscopy showing that MYC sat in nuclear pores. And then if we think about the periphery of the nucleus, so it's made up of these dense heterochromatin domains, which are associated with lamin or lamin-associated domains, which basically contain repressed chromatin. But then at each nuclear pore, there's a channel which is composed of euchromatin. And it's these channels that are where the chromatin is poised for rapid response to signals from the tumor microenvironment. And this is where MYC gets recruited when signals come in to activate it post-translationally. And recent work has been asking what is different about the chromatin that sits at these nuclear pores. And one of the things that's been shown is that it's a site of what is called epigenetic memory. And so once a gene is induced and opened at the nuclear, localized near the nuclear pore, it's remembered and it's kept somewhat active by these nut proteins, these nuclear pore complex proteins. Keep them open, keep this open, so that the next time the signal comes, the gene can be activated much more quickly. And our data shows that MYC is involved in this. So this work was driven by a very talented graduate student who's gone on to a postdoctoral fellowship with Bob Eisenman. And he used proximity ligation assays where you can amplify a signal if two proteins are within 40 nanometers of each other. You have these two antibodies, and the antibodies contain primers, and you use PCR to amplify up a signal. And it only happens if the two proteins are within 40 nanometers, approximately 40 to 60 nanometers of each other. And so he looked at proximity ligation between MYC and these different nuclear pore proteins. And he found that TPR and NUP153, which are the proteins that are part of the inner basket of the nuclear pore, interact with MYC, whereas these other nuclear pore proteins don't. And he showed that phosphorylserine 62 MYC is particularly enriched with its interaction with the nuclear pore. And we used, with Xiaolin Non, we used super resolution microscopy, and we showed that while total MYC, this is the distance from a nuclear pore, total MYC has a peak that's around 60 nanometers, and then it goes off, but then there's total MYC throughout the nucleus. Phosphorylserine 62 MYC really just peaks at about 60 nanometers from a nuclear pore. So the other thing that Yulong did was look at this temporally, and he's using manipulation of pin 1. He also manipulated phosphorylserine 62. Either one will prevent it from localizing to the nuclear pore. And this is just to show you temporally, if you activate MYC with Mitogen, you phosphorylate it, and pin 1 recruits it to DNA. And this happens in the wild type, but in the knockouts, this is greatly delayed. And if we use ATAC-seq, so this is going to look at the openness of chromatin across the genome. And you can see that if we take wild type cells and starve them and then stimulate them, we have a lot of genes that are opened and closed. If we do that without pin 1, from starved to stimulated, there were 0 statistically significantly open genes at this early time point. So really the rapid response to a mitogen stimulation requires MYC to be phosphorylated and recruited to the nuclear pore structure. So this is just a summary that a new student of mine wrote, where he is showing both the temporal and the spatial regulation of MYC, and that this is involved in the rapid response to environmental signals. So now I want to bring up something that's a new direction. So now we're really interested in, because we keep finding that MYC is highly associated with replication stress pathways. And so this actually fits really well with Elda's talk. In that MYC is both a driver of replication stress, as is RAS, because they're upregulating proliferation, and that causes a lot of stress. But MYC is also a mitigator of replication stress, and it plays a role in DNA repair as well. And what this is a recent review showing that some of the steps that are involved in replication stress. So replication stress can be caused by nucleotide deficiency, the collision of highly transcribed genes with replication forks. And when this happens, repair proteins become sumolated, and sumo signals are really important for localization of proteins to different regions in the cells. And in this case, upon stalled forks and collapsed forks, the repair proteins like RAD51 become sumolated, and this recruits the stalled and collapsed forks to the inner basket of the nuclear pore, where actually there's a liquid-liquid phase separation event. So very much reminiscent or reflecting what Elda was talking about. Now, one thing I'm wondering about is whether there's ever stress granules in the nucleus. But anyway, there is a phase separation that happens at the inner basket of the nuclear pore that people have described. And this is probably important for the rapid enzymatic events that need to take place to then repair the DNA. And this involves a destimulation event. So you first need to get stimulation and then destimulation. And this is what helps repair the DNA. And we had previously shown that SENP1 plays a critical role in controlling MYC, in that destimulation of MYC stabilizes it. And also stimulation, another group had shown, controls PIN1 activity, where it also, SENP1 destimulation also activates PIN1. And what we showed is that SENP1 and phosphoserine 62 MYC are localized to the nuclear periphery at these nuclear pore structures. And then my new student who's taking on this project, he's really interested in not so much the transcriptional control of the nuclear pore, but really this DNA repair that MYC might be involved in at the nuclear pore. And he's shown that if you kind of enhance DNA damage with PARP inhibitor treatment, you increase the recruitment of MYC to the nuclear pore by PLA. And we had also and this is using low passage patient-derived cell lines from PDAC patients. We've also shown that there's a correlation with olaparib sensitivity and the level of the MYC-G1 transcriptional program. So now back to MYC and PDAC. So I don't know, this is kind of going without me. Okay. As I told you in the beginning, KRAS post-translationally activates MYC. We know this is an early event. This is some phospho-62 staining that we did a while ago, where you can see from normal to PANN up to PDAC, you get increase in phosphoserine 62. And this is important for driving the plasticity and the tumorigenesis transformation of the tumor cells themselves, as well as the desmoplastic reaction. And if you just simply look at TCGA, poor high MYC or MYC alterations is associated with poor prognosis in PDAC. And we've been following this up with a really large RNA-seq data set that we've created with about 300 patient samples. And this is work being driven by a talented physician surgeon, Patrick Worth, and a research assistant professor, Jason Link. And this is just looking at some of the samples in our data set where Patrick is calling something a rapid recurring PDAC, which means that it recurred within 6 months post-resection, which is a really devastating event for surgeons who are who think that patient's resectable, but then they rapidly recur with metastatic disease. And then we're comparing these to long-term recurs greater than 18 months. And this is a density plot showing you the GSVA score for the Hallmark MYC-V1 pathway. And you can see that the rapid recurs are in blue. So they tend to have a higher MYC activity score than the long-term survivors. And then bioinformatic work by Carl Pelz showed that we do have the MYC pathway is enriched in the rapid recurs, which are in red, over the long-term survivors. So, and interestingly, we noted that whenever we have the MYC activity high, we also have these replication stress pathways being high, as well as DNA repair pathways being high. So both MYC as an inducer of replication stress and a mitigator through DNA repair pathways that all get upregulated at the same time. And this is some staining that we've done on some of our patient samples. We've done by a talented research associate, Colin. And what he did is he did some cyclic immunofluorescence. And what you see here is rapid recurring patients at very high levels of phosphoserine 62 in MYC in green. They often tend to also be CK17 positive, which is a sign of the basal phenotype. And we often, when we see high MYC, we see high phospho-RPA, which is a sign of replication stress. And the long-term survivors just have much lower levels of MYC. So using our full data set, which comprises 225 primary tumors and 79 mets, we find that if you look at the distribution of the Hallmark MYC GSVA score versus the replication stress GSVA score, we see about equal numbers of samples that are high in both versus low in both. If we look at just mets in our database, we see more of them have high MYC and high replication stress. But one of the things we observed is this is particularly prominent with liver metastasis. So to try to model this, we've created a new mouse model, a new genetically engineered mouse model, where we have an inducible Cre, so we can turn it on in the adult pancreas, acinar cells. And then we have the typical lock-stop-lock KRAS G12D, and then we're driving low but deregulated levels of MYC from the Rosa promoter with a lock-stop-lock. So in the present, when we turn on Cre in the acinar cells in 6 to 8 week old mice, pancreases, we turn on RAS and we turn on this deregulated MYC. And you can see that the control mice and KC mice and MC mice don't come down up to 700 days with tumors, but either a het knock-in of MYC or a homozygous knock-in of MYC. Now you get accelerated tumorigenesis, and we see ADM, PANIN, as well as poorly and well-differentiated PDA. Interestingly, these tumors follow some of the similar genetic events that happen in humans. We lose P16 expression early on, and we lose SMAD4 expression, and we inactivate P53. And this has just been more recently shown when we're comparing here KMC, which is the name of our mouse model, ERT2 to KPC. And you can see KPC, as others have reported, they don't lose SMAD4 or P16. Unlike our model. So we've also looked at these tumors using Lisa Cousins' multiplex IHC platform, which she uses 22 different antibodies to enumerate the immune phenotypes in tumors. And this is just an example of some of the images that we got from this, where we're looking at immune cells and epithelial cells in these tumors. And one of the things we've done, and this is preliminary, analyses, but it looks to us like there's some differences, particularly in DCs between KMC and KPC. And I've talked to some other people who have also observed this. KPC really virtually have no DCs, dendritic cells, whereas KMC have plenty of dendritic cells. Both mouse models don't have very many CD8 T cells. There's a little bit more in KMC. Otherwise, they're pretty similar. KMCs have a little bit less macrophages. And we're basically trying to model, this is from Lisa's data, this is therapy naive primary PDAC human. And we're trying to get a mouse model that can mimic this as closely as possible. So one of the other nice things I'm excited about with our KMC model is that we've made cell lines from these. And what's really cool is we can take these cell lines and orthotopically inject them. And the PDAC that arises is actually has a large stromal invasion and is highly desmoplastic, very much like the autochthonous arising tumors and like human tumors. And this is work by Isabella, a graduate student, and Patrick, the surgeon. And they've gone on to show that this mouse model is resistant to standard of care using just single agent gemcitabine or the combination chemo FOLFIRINOX. We've also shown that in the autochthonous model that mice that had metastases had a much higher transcriptional signature for MYC and replication stress. So similar to what we showed in human. The other nice thing about this model, unlike some of the KPC lines that we've tested, is it's reliably metastatic. So after orthotopic transplant, we can see by ultrasound the appearance of mets in the liver, and that's just shown here, and they look very much like human mets in the liver. And so we're excited about this mouse model as a way to study, to test therapies. So just a couple of last slides. Different ways of targeting MYC. This is a perspective written by a postdoc, a past postdoc in the lab. Different ways of controlling MYC transcriptional regulation. There's BAT inhibitors, there's G quadruplex stabilizers. These things would all affect MYC gene transcription. And then post-translational regulation, things we've been working on. We've been working on a drug that activates PP2A to dephosphorylate serine 62 or PIN1 inhibitors to prevent PIN1 from helping recruit MYC to DNA. And so I'm just going to show you a couple slides on the PP2 activator. So we're excited about this because PP2A is a very potent tumor suppressor, controlling many pathways, including MYC. And recently, our collaborators who developed this small molecule PP2 activators, these SMAPs shown here in yellow, had a cell paper in which they demonstrated that this one of their small molecules was really specific. So even though PP2A makes a lot of different holoenzymes to control a lot of different pathways, the one that controls MYC contains the B56 alpha B subunit of the PP2A holoenzyme. And this one compound nucleates that formation specifically of the PP2A B56 alpha. So this is this compound DTO61. So this is an invited spotlight that we wrote on this small molecule, and it just shows that normally in cancer, PP2A is actually downregulated, and MYC is activated by phosphoserine 62 to do all of its stuff. If you treat with this small molecule, what it does is it binds into this pocket and stabilizes the trimer that targets MYC. And this leads to MYC dephosphorylation and degradation. So we've shown that past postdoc Megan Jolly had shown that treating with DTO61 and like the PANC1 cell line decreases MYC expression, combines well with gemcitabine. A past postdoc, Brittany, showed that if we treat mice that have been given cerulean to induce ADM, we can, if we treat with the small molecule activator DTO61, we suppress acidoductal metaplasia and the pancreases look more normal. And then in a treatment that we did a while back with the KPC mouse model, we actually get a pretty dramatic increase in survival in the autochthonous KPC model. And VD postdoc in the lab demonstrated that this is associated with dephosphorylation of MYC. So to summarize, what I've really kind of tried to tell you about is how MYC is really, it's not all about like whether MYC is gene amplified or whether it's just expressed. It's really, you have to think more about its post-translational activation that really controls its temporal and spatial activity. And one of the things, not only that I wanted to show you from some of our human studies with our human data set is that MYC is post-translationally activated in rapid recurring patient samples that are liver tropic, that have high replication stress pathways, and MYC both would exacerbate this, but also mitigate it through activating DNA repair pathways. And that we've created a new GEM model that can recapitulate this with reliable 100% metastasis to the liver, high replication stress and signaling pathways that we can use to try to do new therapeutic testing and some strategies to target MYC, which we want to think about in combination probably with immune checkpoints and other things to try to target. And lastly, I want to thank all the people in my group who did all this amazing work. I tried to show their pictures and mention them along the way. Our great collaborators, in particular Lisa, who's done the immune phenotyping for us, and external collaborators and my funding sources.
Wow, thank you so much. I feel like I've just learned so much over the past 20 minutes. So some questions from the audience that I think fairly straightforward. Do you think that NP152 could be a limiting factor for MYC-dependent transformation? So in other words, if we look at patient outcomes, does the abundance of MYC and NP152 correlate in combination with patient outcome?
Yeah, so you mean the nuclear pore protein, I think is what they're talking about. That's a really good question. Nuclear pores are really very cool structures. I think they're very much like these stress granules, really these kind of sites where you get these liquid-liquid phase separation and you can have a lot of reactions happening at them. And people have shown that you get about a 10-fold increase in nuclear pores in tumor cells over normal epithelial cells. But I've not looked at whether that's the case. And I don't know of anybody who's trying to drug the nuclear pore. I think it would be really interesting to think about drugging liquid-liquid phase separation or something like that, like what Elda was talking about.
Actually, one of the PIs here is looking at drugging nuclear pore complex, Dr. D'Angelo. Yeah, so that's
very cool.
One of the reasons we'd all be interested in hearing about that.
Yeah, very cool.
in context of the DNA damage and replication stress that you were talking about, so we know MYC does different things in the p53 wild type versus p53 mutant context, right? And so do a lot of DNA damage repair proteins. So do you think that relationship between MYC and replication stress is driven by a p53 mutational context, or do you think it's also independent?
That's a really, really good question. I mean, more than 50% of PDAC is mutant P53, right? And that's a really great point. We have not parsed that out when we're looking at, like, when we're doing rapid recurs versus not, or lung tropic versus liver tropic, and some of these things that we've parsed our data with. We've not, I've not done that. I've not asked that question. That's a really, really interesting question. And you're right, because, you know, I don't know how much of that would control mix activity at the nuclear pore. We have not looked into it. It's a really good question.
Another interesting question from Jeff, which is a question I always wonder about, actually, because, you know, in your mice studies, you show that the DTO61 prolongs survival, but really it's only for a bit and all the mice end up dying. So is this because there's only partial inactivation of MYC with the P62 dephosphorylation, or are there escape pathways or resistance, or what's happening there?
Yeah, it's a really good question. When we've looked at tumor growth, so we've done this both in our, we have a triple negative mouse model, GEM mouse model, kind of using the same strategies as our PDAC GEM model. When we look at tumor growth, it's like shut down. We can look at it by ultrasound. It's like shut down for like 20 days. And then it slowly starts creeping back up with DTO61 treatment. So we haven't yet caught the right time points to try to figure out what switches it. We have done some studies in the TNBC model combining with a checkpoint inhibitor, and that actually works a lot better. We've also combined in the with olaparib in the triple negative model, and that actually works really well, and the tumors shrink. So there's, I think, I don't know if there's some way of bypassing it. There's definitely PP2A, you can develop mutations that make it resistant to the drug. You can upregulate other subunits that would make it resistant to the drug. So there's definitely resistance mechanisms to the drug. And I feel like, as with kind of most things, we're going to need combination therapy.
Yeah, actually, and that brings up a really great question, I think, for this whole thing. So I'm going to ask Elda to come back on as well and turn on her video and audio. Listening to both your talks, I feel like one of the things that's really great is, like, these are all new things that we're finding out about PDAC, right? And it is a really horrible, I mean, all cancer is horrible, but it is one of the more horrible cancers. And so, as Jeff was asking earlier to Elda, but I think it sort of relates to both of your work, is what do you see the future of therapeutics in PDAC? Like, it is really desmoplastic, so can we get inhibitors into them? Like, how effective are all these new things that we're finding out going to be as therapeutic targets?
Yeah, I think it's a good question. I'll just start out just briefly, but I'm sure Elda has like some really great thoughts about this. You know, there is this whole question about like what gets in. But one of the things that I am constantly reminded of is our patients, even though they may have what looks like a really desmoplastic tumor, the ones that respond to like FOLFIRINOX or something like that, they still respond really well. So drugs can get in there, even though it's very desmoplastic and has a high interstitial pressure and all that. But I think that you kind of need to start that process. I mean, one thing that we've observed is that if you inhibit MYC, you start that desmoplastic tumor microenvironment starts to collapse. And so we are able to get small molecules in there. I think you have to kind of initiate the cascade, but you're going to have to keep hitting it with combinations. And whether they can all come at once or they have to be sequential, I think that that's something you need to understand. And then you have to, once you can start getting immune cells in, you're going to have to really, you know, activate, figure out how to activate those T cells. Go ahead, Elda, sorry.
I mean, I think I agree with a lot that Rosie's saying. And I think one for me at least, I mean, where I've decided for us to focus our work is, I think we do have to look for shared properties or shared mechanisms. And I mean, this is why the stress adaptation is appealing to me as a process. So, you know, obviously all the cancer cells, all the components of the microenvironment are under stress. They all need to adapt, right? And I think also, you know, the crosstalk between cancer cells and the other components of the microenvironment, be some of the work that Rosie was talking about, work that a lot of people in the field are doing, the work that we're doing on how the cancer cells can affect stress adaptation of the non-cancer cells into a microenvironment and vice versa. And I think those commonalities, or looking for those shared mechanisms, I think can provide a better window. And for me personally, I mean, I think there is a lot to do in chemo prevention, right? Or detection, right? I think what I saw that my work can contribute is in the chemo prevention aspect. And that's why we went for models like obesity. And these are certainly modifiable factors, but they're not so easily modifiable in real life. But having biomarkers or having means or just targeting this or blocking this from occurring or delaying it, I think that is, you know, something to think about also as a field, right, in this type of strategies.
That is so interesting you say that, because I feel like both with the stress granules and all these, you know, post-translational modifications in MYC, we're talking about really dynamic processes, right? So how much do you think that things like, say, obesity or other stress, environmental, lifestyle stress factors, how much do you think reversing those behaviors could actually, even after you get cancer, say, how much do you think it could affect how well you respond to treatment, or, you know, how well you do basically on whatever standard care we're giving people?
I guess I think it depends on mechanism, right? How are these changes occurring? I mean, I think for epigenetic modifications, perhaps you can argue that once there, I guess Rosie can probably speak better to this, like how, I don't know how lifestyle changes would revert that process. I think and you know, we talk about this. I want to be cautious about this aspect of obesity as a modifiable factor. It is and it's not. You know, it's not necessarily modifiable. It's something that I think, you know, we have to consider that this is part of our population and how we can help prevent the consequences of this. Of course, combined with modifiable approaches if possible, but it's not always.
Yeah, I don't know. I mean, I think that once you have this, you know, really aggressive tumor that has these intrinsic features of activated MYC, and you know, it's mitigating the replication, the stress pathways that are being activated by the tumor microenvironment, and you've got stress granules that are helping mitigate those also. You know, those tumor cells have become really plastic and able to handle lots of stress. And so it's going to be really, really hard to, and I think that their epigenomes are plastic too, you know, at least in a lot of cases, particularly, you know, I mean, I found it so fascinating that the nuclear pore is a place where, like, why do the stalled or you know, collapsed forks go there? Why, you know, what's going on with that? And I think a lot of it has to do with, like, those EM pictures and some of the stuff Claude's done too has really shown these areas of you know, where you could get this phase separation, where there's space for all these reactions to happen, and that's where things have to happen. And if we can figure out how to, you know, maybe what we need to be targeting is dynamics or state change or phase separation or something that is allowing the cells to survive under all of this stress and pressure. That I think, you know, and whether modifying something, if you could modify something like obesity or something like that, you know, I don't know, you know. Then we also have to think about things like modifying cachexia and stuff like that. And nerve innervation can affect all of this stuff. So I think we have to take a very broad approach to all of the heterogeneity of the different types of cells there and how they're all working together to try to support this cancer, unfortunately, you know, to allow it to survive all of these different stressors that it's encountering.
Yeah, that's such an interesting idea, that going through all that stress is basically making sort of an uber cancer cell that's up to anything you throw at it. I know Cosmo had a question as well, so I'm going to let him get his in.
Hi, Rosie. I just
Hi.
wanted to ask whether there's any evidence for contextual role for MYC activation or MYC expression. So for example, do you know whether there's differences in PDAC subtypes? And also the nice data in the liver, do you know whether other metastatic sites would, or would they be, do you expect it to be the same as the liver?
It's a really good question, and this is all unpublished data that Jason Link has been doing. And actually, he has a talk at the AACR Pancreas. So I encourage people, you all, to watch it. You know, he's been looking at the difference between liver and lung tropism because we have this big RNA-seq data set with really high clinical annotation. And he's found actually a gene signature that is organotropism that will predict whether you're going to go to whether the tumor from the primary, the signature from the primary, whether it would go to the liver or lung. And one of the hallmarks of that is this high MYC, high replication stress that I was alluding to. And so why is that? Like, is that because other places don't have as much stress, and so the ones that can survive in the liver have to have that? Or is it that, I mean, because you might say, well, we know that in the liver that's high because maybe there's just more stress in the liver, but we're finding that actually the primaries that can go to the liver, those are the ones, they also already have it at the get-go. So is that because those cells are just more able to survive there, and that's just a worse place and an easier place to get mets? And so if you have that signature, then you can get mets there, and then you know you're going to have a poor prognosis. We don't really know. I guess you know, we're trying to learn about this, and we're hoping that the multiplex IHC and the cyclic immunofluorescence, you know, to really look spatially and cell contextually. Now that we have this phospho-specific antibody, we're hoping to be able to look at activation of MYC and what the cells are around it. I see in the chat, Tony had a question about whether our phospho-62 MAB antibody. So we don't know the answer to that. When we had a couple different polyclonals that looked like they were, one was specific for cis and the other was not. But it's been really hard to prove that. And we have, for our monoclonal, we haven't figured that, we haven't been able to test that. We you know, the tests we've done is we know it doesn't react to serine 62, alanine mutant MYC, and we know that even if we treat tissue sections with, or if we do cross with a MYC knockout, it doesn't react. And then also if we treat tissue sections with a phosphatase, we get rid of the signal. So that's the extent so far. I mean, we feel really good about this antibody, but I would love to know whether it recognizes cis or trans. And if anybody had a really good assay for that, I'd like to know. We've tried like using the mimics, the proline mimics, and making synthetic peptides with proline mimics. And the proline mimics just aren't good enough for an antibody. Antibodies are too specific.
Thanks for answering that, Rosie. I was wondering how to get it into the flow of questions. I'm gonna end with Jeff's question actually, because I think there's a bigger question there for both you and Elda, in terms of, you know, this idea that what is the right, do cells have to be replicating for all of these targeted therapies to work? And I don't know, Elda, if with the stress granule inhibiting strategies, is that going to be different? Because I do think for a lot of like MYC targeting, maybe it does need to be proliferating. I don't know, Rosie, maybe that's naive, but what do you two think about that?
I guess who goes
Elda?
first? Okay. If the question is specific with the stress granules, I'll say that there will be a difference on whether the cells are proliferating or not, or at what stage even in the replication process they are. That's work that I didn't get a chance to talk about today, but it's you know, it's something we're very excited about. So that will matter. I mean, again, you know, we're talking just different degrees of sensitivity and different degrees of, you know, accessibility. So I think it is, in the context of the stress granule specifically, it will make a difference. And it's something that we have to think about, you know, like therapies that may perhaps induce a particular type of arrest. You know, what do we combine that with, right?
That's so interesting. I'd have thought with the stress granules, it would actually be proliferation phase agnostic, because most of your phenotypes are apoptosis, and
yeah,
so
we were very surprised. Yeah.
So it was, sorry, so it was both G1, so it was G1 and S, or when was it?
So do you mean the question?
Yeah, so I missed what she said about which phase it was. I think it cut out.
I didn't specify it. I was saying that I think it is phase phase dependent. Yeah. But I think, these are things that did really get me thinking also, the therapies that we are using. I mean, in this case, we happen to find that, because we were looking for that, but how much do we consider that really in everything that we use? Like we have, let's see, even chemotherapy, right? Fulfurinox and all these things, they probably do induce the cell cycle arrest in the cells that they've reached. Right. So how does that really, if you're combining that with other therapy, how does that factor in? I don't think, to my knowledge, that's been explored very much. But I mean,
a complicated question, yeah. But I think a very clinically relevant one.
I think clinicians perhaps in the audience would know better than me, but I feel like that hasn't really been addressed. Yeah,
I think it's very complex. For MYC, it's like MYC needs to be on for the cells to take on a new state, but it actually has to be turned off for the state change itself. So it has to be all kicked off of the super enhancers that are defining one state, and then they can rewire, and then it has to be able to come back on to allow that state to get fixed and start proliferating in. So that's a really complicated dynamic that we think, post-translational control is involved in, but we haven't gotten that whole thing figured out. So you could imagine turning MYC off, maybe it can rewire its chromatin, but it can't fix it or turn it back on or start proliferating again. And so I guess in that case, it would still work. But I think your question about, therapies and how they might affect, a lot of the targeted therapies we use, right, stall the cell cycle, and what will that do to chemotherapy? In a lot of cases, we still see that there is this combination with chemotherapy. And I don't know why, because you'd think if you stalled it, then it wouldn't work, right? But then the other thing to think about is, why would those targeted therapies, at least the cell intrinsic, things that target MAP kinase pathways and stuff, you would also be worried about them stopping T cell proliferation and stuff like that. And yet when we treat with DT-061, we actually see the tumor cells arresting, and we see an influx of immune cells. And so you're kind of wondering how that's happening. And so I think we have to be really cognizant of not just how a drug is affecting the tumor cells, but also how it's affecting the whole tumor microenvironment. And the immune cell influx. And so anyway, it's a very complicated thing, and I think that we need to understand how tumors evolve. And one way to do that would be with window trials. We have a window of opportunity trial right now for patients with liver mets, where they come in, they get a biopsy, they get 10 days of olaparib, or a MEK inhibitor, or a BRD4 inhibitor, or a WEE1 inhibitor, different things. And then we get another biopsy in 10 days. So we're not really in that time looking for a, effect on survival. We're looking at how the cells rewire, and then we're throwing everything at it, transcriptional, proteomic, all that kind of stuff, to try to understand how they rewire. And it's actually striking how much just in 10 days a tumor really rewires. And then if we can understand that and capitalize on it, I think we could, and I think maybe if we can capitalize on this on this stressed phenotype, like Elda's talking about, right? Since we know that can give you a therapeutic window, if we can capitalize on this stressed phenotype.
I was gonna say we should end on that hopeful note, and that's what Jeff said as well. So I guess. Elda and Rosie, can you please stay on for the trainee roundtable? And if everyone else, except the trainees who are staying for the roundtable, you stay on too. If everyone else can go back to their Monday mornings after that, really, really fun session. Thank you so much.
Thank you both.
Thank you.
Thank you.
Thank you. It's super interesting that the nuclear pores. I mean, I always found your work with MYC and the spatial transcription there. That was always just fascinating for me.
Yeah, I was really excited about it just in terms of how it could control poised genes. And then when I started learning all about this, collapsed forks moving there, DNA repair happening there, all of this kind of stuff. And then, it was really, I went through it really fast. It was all kind of new stuff. And so I was really worried I wouldn't be able to present it comprehensively. But that whole sumo ligase that's involved in repairing the stalled forks that also controls MYC and MYC's ability to interact with the nuclear pore, you just start like putting all these puzzle pieces together and thinking, wow, it's playing more than just a role in just transcription, and Martin Eilers has talked about that too, this idea that MYC might be helping to resolve R loops that happen at promoters. So there's been some hints of this. So yeah, I was wondering if I shouldn't call it a transcription factor anymore, if I should call it like a master regulator or something. But
it's fascinating and scary at the same time. That
do you ever see stress granules? I mean, so how do you distinguish what's like these non-membranous granules, because they happen in the nucleus
what's
too.
A stress? They do. So I think the broader umbrella term is condensates, right? Because like anything that phase separates, and I think depending on composition and under what, I think it's called this or that. So granules generally are thought to be cytoplasmic, but there was some, and the mechanism sometimes, usually the nucleated proteins, they tend to be different, I think, but I'm not actually sure that that's really carefully been shown. Like, I don't know if there's a study showing that, G3BP, for example, is only stress granules and nothing else. It's not to my knowledge, but, I don't know. But
well, it could be different degrees of kind of the same idea, these condensates that
I think absolutely.
Intrinsically disordered proteins coming together, causing the condensate, and that allows for much more rapid enzymatic reactions to occur for repair or whatever needs to happen, right?
I think absolutely right. I think the one unifying feature is that the intrinsically disordered regions in proteins, and then so basically anything that is multivalent. So that's why RNA, a lot of these are RNA granules, because RNA is a perfect multivalent molecule. Right. Exactly. If they wanted anything to be able to bind with stuff, that's it. And if you look at the granules, the ones that are preferentially recruited, they are longer, the long non-coding RNAs, because they're just longer and it's more multivalent. But I think, that's the criteria. IDR regions tend to do that and provide that flexibility for that. So any protein that has that could be, in theory, a nucleator once it reaches a certain concentration.
Yeah, super cool.
I mean, it's super exciting. And it's great that there's more, a lot of people are working on it right now. And there's a lot of more tools that you can kind of start to address some of these questions. Because I was always, in my mind, I'm just like, how do I link this causally? Like, how do I say it's the granules and not anything else that you would be perturbing with any drug or genes or anything like that? So I think now we're in a spot that we can start adding this information that we've gathered from, other people that have published beautiful work on, the different domains and the IDR regions and things like that to link it. And we can do all the bigger studies and figure out exactly what is downstream that they're regulating.